So we're going to talk about just our defense mechanisms, B cells, T cells, and immunity, real fast. This is the only meme I actually created. All the other ones I just steal offline. This is the one. If you get it, it's funny. If you don't, watch Dave Chappelle. It's funny. Okay, our defense mechanisms. We have to be able to protect ourselves from the outside environment. Okay, they're called pathogens from the outside coming in. We have two different types. We have innate, which is what we're born with, and we have adaptive, which comes from exposures. So vaccines is a adaptive immunity. If you breastfeed, it's adaptive immunity because you're giving IgA from the breast milk to the infant. It's adaptive immunity. You're giving them the antibodies. Cells, they all come from the same precursors inside the bone. If it goes to the thymus gland, it becomes a T lymphocyte. If those things stayed in the bone and it developed into a B cell because it comes from the bone. So B cell comes from the bone, T cells mature in the thymus gland. Easy to remember those two things. Okay. So innate immunity, things that we're born with. We have external. Remember, there's anything that can get in from the environment, we have to be able to protect ourselves. So our skin technically is the largest organ we have with our entire body, and it's protective. It keeps things out. Digestive tract, respiratory tract, and GU, those are all other areas and ways for things from the outside environment to get in. So we have to have some protection there as well. Digestive tract, high acidity of the stomach, kills anything that gets in the stomach. Respiratory tract, your mucus traps all those pathogens in the mucus, and then you cough it out. And if it's like yellow or gunky, you know you got a lot of crap in there, you were sick because you have all this stuff you're coughing out. Genitourinary tract, acidity of the urine, supposed to help prevent UTIs and so forth. Internal, these are stuff within our cells or different structures that provide immune function. Phagocytic cells are basically just floating around in our bloodstream, scavengers eating up different things. Interferons, anytime you see the word interferon, just think of viruses. They always go together. Interferons work with viruses. We have complement proteins, endogenous, natural killer cells. These are transplanted tissue cells. Mast cells release histamine. What does histamine do? Makes you leak fluids, so you start getting snot running down your nose. So when you think of mast cell, think histamine. It causes swelling to happen in your joints when you get an injury. Okay, histamine causes the capillaries to become very leaky. They get in there. Again, we went through taking an antihistamine. This makes it so you don't have a runny nose. It stops the leaky. Oops, I don't want to do that. Okay, so innate immunity again. With phagocytosis, neutrophils are the first ones to come. So for infection, responsible for bacterial and viral, so your neutrophils. Then your monocytes show up. And you also have our innate organ-specific ones. Liver has filtering cells. Your liver is perfectly functional. You don't need to put those little stickers on your bottom of your feet. They're not going to do anything. Your liver is perfectly normal, can clean everything out. And again, those phagocytic cells in their location, Kupffer cells are in the liver, alveolar macrophages in the lungs. Remember we talked about microglia already? Those are the immune system and the nervous system, immunity in the nervous system. And here's just examples of the slides of what they look like. Here are the interferons. Remember, these are always doing with antivirals. So just remember, interferons have to do with viral infections. Okay, adaptive immunity, things that we're getting. Okay, we have to defend against specific pathogens. Okay, we have to know what we're fighting against. Basically, immunity, think of it as a blueprint. It's a method of defeating an enemy. And they just give us a blueprint, and when we get exposed to that pathogen, our body already knows the plan, and we can produce antibodies. So it's mediated by the antigen, which is the pathogen, and antibody formation. So B lymphocytes, they come right from that bone marrow we talked about. Bacterial, some viral, but mainly bacterial infections for those B lymphocytes. These are the ones that are responsible for making antibodies. Only the B lymphocytes make antibodies, and they are specific to a pathogen. So they know how to fight it off. We have different infections, we have to fight them off differently. Very short lifespan. It's also called humoral immunity, from the humerus, the bone, humeral bone, same thing. Or antibody-mediated immunity, because it's antibodies that are fighting off the infections. So, if you look at this picture here, okay, we have a developing B cell at the top. Then you'll see it has different B cells, and they have different receptors with different colors, which means they are specific for certain pathogens. Only certain things will fit in those slots. If that pathogen is in your system already and it's floating around, it's going to bind to that B lymphocyte. And when it binds, then it starts replicating and producing antibodies. And all those things at the bottom are all the antibodies to fight that specific pathogen. Okay, it's all specific. So that's what the B cells do. Now, how do the B cells know what to do? They get help from the T lymphocytes. So the T lymphocytes, remember, it goes to the thymus, and they become T lymphocytes. T lymphocytes attack host cells that become infected with a virus or fungus. Transplanted human cells, so whenever you get a transplant, you have to go on high-dose steroids to knock out your immune system. Because if you don't, those killer T cells are going to go to that transplanted organ and kill it, because it sees it as a foreign body. Okay, so that sometimes that can happen as well. So a lot of people with transplants will go on those high medications. Cancer cells will do it. T lymphocytes don't produce antibodies. It's called cell-mediated immunity. So T cells are cell-mediated. What does that mean? I think it's the next slide. We have two different types. Okay, this is a comparison, just like a summary slide. Read that on your own. Do I have it here, or is it down more? It might be down more. Okay, we'll get to it. We're kind of explaining it right now. Okay, so our lymph glands. We have our lymphoid organs. You guys, for PAs, we're already palpating all the lymph nodes. Obviously, lymph nodes makes all your lymphocytes. So when you are sick, they get bigger because they have to produce more and more cells, so they pop. So when you're sick with a bacterial infection mainly, and you start feeling for your lymph nodes, they're going to be big because they're fighting off an infection. They have to get bigger to make more and more cells. They also have other ones, the thymus gland, which we know spleen and lymph nodes can also store and make stuff as well. So your spleen is there as well. Secondary organs, spleen, thymus, bone marrow, lymph, and so forth. Tonsils are here and whatnot. So these are all secondary lymphoid organs. Don't go into great details with these things again. Just know that they're there. And our lymphatic system helps prevent edema. Okay, it just takes excess fluid out of our blood system, out of the interstitial space, and puts it in the lymphatic system, which then recirculates in our body and goes somewhere else. They think that lymphatic blockages is causing some cancers to form because cancers can get out and we drain it through the lymph system, but if your lymph system is blocked, cancer cells have nowhere to go and they start accumulating and then they can embed somewhere and cause cancer. Okay, so B lymphocytes. Big thing with this one, they have to be exposed to the specific antigen, then undergo cell divisions, and the main goal, produce antibodies. That's the gist of what you got to know from this slide. They get a specific antigen. So here is a helper T cell. The helper T cell, all it does, floats around the bloodstream, If it sees a pathogen, it grabs onto it, and then it presents the pathogen to the B lymphocyte. So the helper T cell brings the pathogen to the B lymphocyte. So now the B lymphocyte knows what we're battling against. Because we've been exposed prior to this, B lymphocytes, they're memory cells. They know what the pathogen is. They know how to defeat it. So they produce all the antibodies to kill off whatever infection is going on. So you need the helper T cells to do that. What disease kills off all the helper cells? HIV/AIDS. So what happens, they don't die from AIDS, they die from pneumonia or some other secondary infection because their immune system can't work. They can't kill it off. They don't have the helper T cells to present any of these simple pathogens to the B cells to fight it off because the helper T cells are gone. And if you, it's not in here, but we'll get to it later on. The helper T cells has the CD4 receptor. So if you ever hear CD4 with HIV/AIDS, CD8 is on the killer T cells. So the proportions of what happens alters your immune system. So this is what happens when our body gets exposed to a pathogen for the very, very first time. So you get your Hep B series, for example, you get 3 of those. So for Hep B. So the first time you get the Hep B vaccine, first injection, you get a primary response. That's just our body. Okay, we have an infection, we have to do something about it, but we haven't really memorized what this pathogen is yet. This is our first time being exposed to it. So we get a small little response. Then some time goes on, and now you need a booster shot or the second series. When you get the second one, now we have a bigger response because this is the second time we've seen this pathogen. Our memory B cells know what to do, and they deliver the response to kill off or fight whatever is going on. Which is why sometimes when especially with that COVID vaccine, if you got the second one, some people got really sick for that day after the second one. That means that it did its job because you got exposed to the virus again, your body knew what to do to fight off and kill it. So it elicits every secondary response when you got that second injection. That response can cause fevers, so some people feel like garbage for a day after getting the second one. But again, that means it worked. That means it did its job. Your body knows what to do. Problems come into play when you get this secondary response is very hyper responsive. That's asthma. That's if you get stung by a bee and your hand swells up really bad. That's a secondary hypersensitivity. People eat peanuts and get anaphylaxis, they can't breathe. That's a secondary response because they've already been exposed to it. Now they get it again, and that's why if you keep repeating these exposures, they get worse every time. So people like peanut allergies. The more times they eat peanuts every successive time, that response can get worse and worse because this response just keeps getting worse and worse with those types of reactions. So these are the types of immunoglobulins. These are the antibodies. There's only 5 of them. Okay. IgG increased after immunization and secreted during a secondary response. So you guys are coming into school this year, you probably needed some sort of titers to see where your levels are for your vaccinations. They were looking at IgG because that IgG means you've been vaccinated and you should have those antibodies floating around. If you don't see the IgG for that bacteria or that type of antibody, then we know you're not covered. You need to get another injection. IgA, external secretions. This is in your saliva and breast milk. This is the way antibodies go from the mother to the baby, IgA, through the breast milk. Saliva in humans, we've kind of lost this trait, but in animals, that's why animals lick their wounds, because they got IgA in the saliva, and they're giving themselves antibiotics, essentially, to the wounds. That's why they lick their wounds. Okay? We've lost it because we take care of ourselves with medicine now. IgE, always associate IgE with allergies. So if you have constant allergies all the time, your IgE will be elevated because of the allergies. IgM, this just means that you've secreted during that primary response. So that first time you got the injection or the vaccine, you're going to produce IgM antibodies because it's your first time exposure. The IgG is your secondary constant response. That's all you got to know for these antibodies. Don't go crazy knowing, just know what each one, IgD doesn't really, we don't really know what it does, but just know it's unknown. As long as you know the other four, you're good. Okay, our T lymphocytes, we have our killer cells, also known as CD8, and CD8 just refers to the type of receptor. Okay, they destroy cells that are infected with viruses or fungus or cancer cells even. So your killer T cells, CD8, are very good at, obviously, killing things. Okay, so remember, T cells are cell-mediated, meaning the cells have to touch. Remember, B cells, B lymphocytes, is antibody-mediated. It just produces antibodies. And the antibodies do whatever it's got to do to kill the pathogens. But T cells, it's cell-mediated. They have to touch. So this is touching right here to kill off that cell that's infected. It grabs onto it, injects its contents, and kills it off. Helper T cells have CD4 as the receptor. And again, it makes the B lymphocytes work better because the helper presents the antigen to the B lymphocyte. So the B lymphocyte, again, knows what pathogen is floating around and knows how to take care of it. So here's another way of looking at it on how they work. Again, you don't have to go into detail all these things. The helper cell is also known as an antigen presenting cell because it presents the antigen to the B lymphocyte. And then your killer T cells just kill the target cell. Yes?
When it says improvability of B lymphocytes to become plasma cells, is that just the maturation of
To make antibodies. Okay. Remember that it has to have the binding of the antigen to the B cell first. Mm-hmm. Then it knows what it is, and then it makes antibodies. So that's a plasma cell? Yep. So the T cell brings it to the B cell to make it. And again, here's another way of looking at it with regards to antigen presenting cells. This is your killer T cell. It's going to kill that, so you get destruction. Here are your helper T cells. Here, that foreign antigen is right there in the middle. It's presenting it to the macrophage or the B cell, telling it what's going on. And here's the B cell, I'm sorry. Presents it, and then it starts making antibodies to dump off. So antigens in summary, we get the stem cell. They can either become a B lymphocyte or a T lymphocyte. Depends on where they mature. The T lymphocyte helper T cell helps the B lymphocyte to make plasma cells, and finally your antibodies. Just remember the T cell, the killer cell, just directly kills it. Killer cell directly kills the infected cell. The helper T cell brings the antigen to the B cell to make antibodies. That's the biggest takeaway from all that. Other types of active and passive immunity, we kind of talked about a little bit about it. Active immunity, this is when you start getting exposures to vaccines. Again, you have your initial exposure, primary response, get another exposure, and you get a larger response. Okay. And that secondary response, maximum antibody production in less time. So we're faster to get those antibodies developed, faster to heal ourselves. Okay, so developing of that secondary response provides active immunity. So once you have that secondary response, then we know we have active immunity. Again, requires prior exposure. We have to have been exposed to something in order to make those the memory cells to make all the antibodies. Also used to make vaccines, and the vaccines aren't virulent, so they can't really cause diseases. All vaccines do, stimulate that primary response without making you sick. So you can use a killed virus like the polio, a live virus which has been attenuated so it's not as virulent, it's not going to cause an infection, Viruses, they can't replicate or anything, so they're kind of just dead, but it just gives us the blueprint that we know if we get this type of structure again, we know what to do with it. And genetically engineered recombinant viruses, and that's your Hep B viruses like that. You don't have to know the details of this, just understand how it works. Passive immunity is giving antibodies from one individual to another. This is breast milk. The baby doesn't make IgA on its own, but the mother can, and it's in the breast milk. Mother to fetus also, while the fetus is developing, antibodies do get into them as well from the mother. And you have artificially with antidotes, snake antivenom. Snake venom, rabies are very, very rare things. We really shouldn't see them very much. So you shouldn't have an immunity to those things because you don't need it. So if you get bit by a dog that you think is rabid or whatever, bit by a snake, they just give you the antibody. So the antivenom is the antibody to the snake bite. And they just give it to you and it takes care of the infection for you. So that's what that is referring to. But again, it's passive because you're not creating the antibodies yourself, you're just giving them from somewhere else, from the mother or from an antidote. And this is just a good summary slide to review. And just, again, generalized, that's it. Just general information about this stuff. Don't go crazy, detailed. Just understand what the B lymphocyte, where it comes from, what does it do. Understand the different antibodies. And tomorrow will be review. Okay? Remember, PA, you're upstairs, 314.